Okay, so once again, we're saying this dichotomous key is to help us identify some of these bacteria. Some of the information I'm telling you is to help your bacteria grow better. A lot of the information I'm telling you is to help us with a dichotomous key that will go from here to here. So, for example, will it grow in fermented conditions? Does it tolerate air? Does it need extra nitrogen? All those things help us identify from a dichotomous key. Okay, biofilms. Organisms live in association with different species. Biofilms, pretty cool. I actually, as much as it's weird, plaque on your teeth, it's actually really, really cool. You will want to brush your teeth after this. Okay, biofilms. It's a complex relationship among numerous microorganisms. They form on the surface of medical devices, mucous membranes, of your digestive system. They form as a result of quorum sensing. So they're basically talking to each other. I'm not even kidding. It's so cool. So quorum sensing is the process by which bacteria measure their density in an environment by utilizing signal and receptor molecules. They're like, oh, how much of there are they? And then it regulates gene expression in response to cell population density. What? It self-regulates gene expression by sensing its own environment. That's crazy. It looks like this. So cool. Okay, stay with me. So you've got your little microbes are vulnerable to the environmental stress. They're all alone. It's so hard. They land on a surface such as your teeth, right? This is your tooth, right? Bacteria lands there. The cell begins producing an extracellular matrix that secretes quorum sensing molecules. So what are those things going to do? They're going to phone a friend, right? They're going to get everybody together and they're going to be all sticky and awesome. The quorum sensing triggers a change in their biochemistry and their shape to make them like stickier and like harder, like a plaque. Then new cells arrive because they phoned a friend. They've been like, hey, come on. We got new species. We got to make some water channels in here. We got to form a hard outer layer biofilm. So we got water fill. And some microbes escape from the biofilm to resume a free-living existence and perhaps to form a new biofilm or other surface. That is insanely cool. Quorum sensing is maybe just the coolest thing ever. Oh, so there it is again. I don't know if this will play. We will try. Oh, it will.
Welcome to the unseen world of survival and behavior. Today, we're embarking on an exploratory journey into the depths of quorum, sensing the intricate communication system among bacteria. Let's unlock the secrets of this microscopic universe.
Quorum sensing is a sophisticated method used by bacteria to communicate. Through chemical signals known as autoinducers, bacteria can gauge their population size and collectively change their behavior. This phenomenon is crucial for understanding bacterial actions from cooperation to pathogenesis.
At a molecular level, quorum sensing is a dance of chemistry and biology. Autoinducers, the chemical messengers, are produced and released by bacteria. As the bacterial population grows, so does the concentration of these molecules. Once a threshold is reached, they bind to specific receptors, triggering a cascade of genetic changes.
Quorum sensing's power lies in its ability to regulate
It's regulating the genes.
It's like a switchboard that controls various bacterial functions. These include bioluminescence in some marine bacteria and in pathogenic bacteria. It controls the production of toxins and other virulence factors.
A critical role of quorum sensing is in biofilm formation. These biofilms are not just clusters of bacteria. They are sophisticated, structured communities that can attach to various surfaces, including medical implants and water pipes, posing significant challenges in healthcare and industry.
In the realm of pathogenic bacteria, quorum sensing is a key player in orchestrating attacks. It enables pathogens to remain undetected at low densities and then synchronize the launch of an infection when they reach a critical mass. This strategy makes them formidable opponents in the fight against infections.
Beyond pathogenicity, quorum sensing is vital for bacterial survival in diverse environments. It allows bacteria to collectively respond to environmental stresses, enhancing their ability to thrive in extreme conditions, from deep sea vents to Arctic ice.
The implications of quorum sensing extend into agriculture and industry.
Okay.
In agriculture, it affects...
Okay, so tell me why we should even care about biofilms. I mean, besides brushing our teeth. Why is this such a big deal in healthcare and everything else? Why do we care so much about biofilms? You can pause it if you want to ponder. This is where I would say talk to your neighbor. So biofilms are on your teeth. They're dental plaque. They can result eventually in cardiovascular infections like septicemia when bacteria from the biofilm enter the blood stream from injuries to the oral cavity. So that's how you get septicemia. Go brush your teeth. Biofilms can also form on medical devices, such as pacemakers, and produce reoccurring infections. Respiratory equipment may also be colonized by biofilm-forming bacteria, contributing more, again, to respiratory infections. And some respiratory conditions like cystic fibrosis can create conditions that permit biofilm growth in the respiratory system. That's not good. Okay, so that's why we care about biofilms. So also, you should brush teeth. Okay, how do we culture microorganisms? This is the part where it's not a selective media, where we're saying, how do you want your bacteria to be happy? So essentially, we introduce inoculum into the media. So we cultivate the microorganisms on purpose to use for research or labs. And there are characteristics of these. I think we did this maybe in the first lab, right? We looked at the shape. We looked at the margins. We looked at whether they're flat, raised, like you look at them like this, you look at them like this, you look at the size, the texture, and all of this is usually part of a key. You didn't have a key, but you will when we do the unknown. Here's some more characteristics, some clinical specimens, and methods used to collect them and to grow bacteria on them. Obtaining a pure culture. Now, friends, when you do the unknown, this is going to be your biggest problem. You think it's not? It is. Separating your 2 or 3 bacteria is a big deal. It is harder than you think. You think you have a pure culture, and then you go back in and gram stain it, and you're like, why do I have a positive and negative? I still don't have a pure culture. You're talking 2 weeks, you will do this, and you will still sometimes have a contaminant in there. Basically, you want a colony forming unit, a single cell or group of related cells that produce a colony. You want one guy so that you know it's a pure representation of the bacteria that's supposed to be in there. So we are giving you a pure culture. We're going to give you one. You will use a streak plate or a pour plate. We are using streak plates. We will do it like this. So basically, this needle will go in your bacteria. Actually, this needle will be flamed. Then it will go in your bacteria. Then you're going to do it here. Then you're going to take a little bit. Then you're going to take a little bit. And hopefully you'll end up like, this is a beautiful plate. If you do this on the first time, I will be shocked. I don't even think my plates look that good. That is amazing. That's like 10 out of 10. Each one of these guys here, that's a single colony. That's ideal. You can also just dilute the plate with broth. It's a little harder. But ideally, you really want your culture to be just one. We're not growing a lot of this other stuff. We're using nutrient broth and agar, as you can see from what you've done. Here's a slant tube. We did slant tubes in lab. 6 types of general media, defined media, complex media, selective media. This is what you need to think about when you are doing your unknown project. Is it selective? Differential is a little bit different, but is it selective? Is it going to actually tell me something? You can request media, and we may or may not say, oh, this is going to be useful. So make sure it's a selective media. Here's some ingredients for culturing, cultural methods, exact chemical composition, nutrients. Usually you've got some beef, soy, and proteins in there.